All right, so it's a great pleasure to be at yet another FishBase Symposium, and I changed my title a little bit because it couldn't fit on the screen. But effectively what I'm going to talk about is how do we recover our fish assemblages, and what insights can we gain on this from body size. So body size is a core aspect of ecology, and there's a wonderful essay by Haldane on being the right size. And when I was an undergraduate, the very first lecture I had in organismal biology, my professor said, imagine the organism as a cube. And he went on to talk about surface area and volume relationships. My PhD supervisor wrote an entire book on the ecological implications of body size and all the things that all the traits, all the way organisms behave as a function you can predict from their body size. I quite like this painting because body size is important unless you're a human because you can eat everything. So I'm going to start with a punchline. I'm going to present our results from a paper that was published earlier this year in Science, where we used body size to look at the twin aspects of proximity to humans or remoteness, and the role of protected areas. And the punchline is basically that benthic fish assemblages can recover when they're highly protected, even when they're near people. But for pelagic species, they really need high levels of protection in remote areas, and that's an important part of the policy debate that we're having on marine protected areas. And the other part of this is partial protection. My question is, why do we bother? My joke is, when did your sex ed teacher ever tell you that partial protection worked? Thanks for a giggle. All right, so our size, so that's the punchline. You already know what I'm going to tell you. So our size-based questions were basically, how does proximity to humans, or conversely remoteness, impact the outcomes of marine protection? Do the outcomes for pelagic and benthic animals vary as a function of the level of marine protection? And do they differ between those two major habitats? And just for Mark, I'm not talking about fish that sit on the bottom only. It's reef associated. All right, so how do we do this? We use baited remote underwater video systems. Effectively, it's 2 GoPros in a frame with a bait arm and a bait basket. This is my postdoc, Andrea Lopez. And what's really important about this is stereo, because as far as I'm concerned, if you're not measuring length, you're only getting part of the picture. So message to everybody, single camera video systems don't work. What do we do then? So we drop them on the seabed, or we drift them in the epipelagic, and we bring the video back to the lab, and we identify and count, but most importantly, we measure. And here you have an example. You can see that you click on the nose and the tail, and the nose and the tail in the 2 synchronized videos, and you basically get a measurement of size down to millimeters, as well as an estimate of error. So what have we been busy doing? At the point that we started analyzing the data, we had 823,849 links from 1,460 species and from 17,411 samples. So that's an individual deployment of a BRUV from all the way around the world. We don't have every place. South America is a gap. North America is a gap. Indian Ocean's pretty good. And we're slowly building up the database. And I just want to acknowledge the collaborators who so generously shared their data as part of this project. So first result. We kind of all know this, but effectively what we did was, here we've got body size on the y-axis, and here are our 1,460 species that are ranked from the smallest. So this is the mean size, and then you see the distribution. So there are actually 873,000 some dots on this graph. And we can see the mean up through here. What we basically confirmed, because we already know it, is that the benthos, we had higher diversity, so more species, and much more abundant. But and I'm going to try and say this, pelagos, because I'm in Greece. In that habitat, we had the smallest and the largest of our individuals. And when you start, if you do a violin plot and the partials to show the probability distributions, effectively the blue, which is the pelagos, looks like Meteora. If you haven't been to the amazing place with the those big rocks with the monasteries on top, whereas the benthic habitats, the size distribution is a bit more rounded and less steep. So this isn't actually confirming anything that we don't know, but it's important because one of our results, which I forgot to put in the slide, but I will come back to, shows that we're actually homogenizing size distributions. All right, so what did we do? We wanted to predict different aspects of size. So we took 3 measures. From the size distributions, we took the first mode, which was the smallest animals, and the second mode of the distribution, which was our large animals. So we've got one measure is small animals, second measure is large animals, and then we also did the size spectra. I'm not game in 12 minutes to explain size spectra, so I'm not going to talk about that. So today I'm going to just discuss the results for small and large fish in those 2 habitats. And then what we did was we used the database that's curated by David Meeuwig from the University of Montpellier, where we have environmental and geomorphological characteristics like seabed depth, chlorophyll, temperature. Then he's calculated remoteness as travel time to market, distance to ports. And then we also know what the protection status is, whether an area where we sampled was totally unprotected, outside a park, partially protected, or highly protected as IUCN 1 or 2. And this is what we found. So just bear with me while I explain this graph. We have, again, body size on the y-axis, and this is travel time. Log travel time in minutes, just so you know, 4 is about 7 days of travel time. The blue lines are for pelagics, blue areas are for pelagic species, yellow is for benthic. The large fish are up at the top in each panel, and the small fish, which is that first mode and second mode, is in the bottom. So in unprotected areas, what you see is that travel time doesn't really make a heck of a lot of difference. So there's not a lot of really refuge for animals in unprotected areas, with the exception of large benthic species. And there we are actually seeing that the further away you get, the bigger the animals get. If we look at the rest of it, what we basically see is that in pelagic environments, small and large, well, we basically see that size always increases with remoteness and protection for both, on average, if you look at it. But what we find is that the pelagic animals are really sensitive to remote areas. So in this final panel, where we see this steep slope, as we get further away in highly protected areas, we really get an increase in the size of pelagic animals, whether they're large or small. What this is telling me is that actually we basically, for benthic animals, we hit a plateau. It doesn't, once you start protecting them, they get there and they don't have to be far away. Now, and this is good, I'm going to catch up on some time for us. Why does this matter? So we have a couple of debates that go on about marine protected areas. The first one is the usual large MPAs are a waste of time because pelagic fish will just swim out of them. My answer to that is, well, then you can catch them over there. And you know, the early arguments in the 70s and 80s about coastal marine protected areas were the same thing. Oh, it's not going to work because the fish will swim out. Well, we have very good evidence from coastal seabed MPAs that, in fact, they have benefits. So, but we're still hearing this argument that large MPAs won't work. But this is showing that they do, and we don't get much bang for our buck for partially protected areas, and that's the problem. And Daniel was talking about it yesterday with paper parks. Australia, for instance, has put 40% nearly of its EEZ into marine protected areas. And we don't really like to talk that much about how much of that is highly protected, right? And in some of our marine parks, you might be amused to know that 98% of them is still open to trawling. 98% of a marine park open to seabed trawling. So we need to knock on the head this question of partial protection. We really need to do that, and I think size gives us an insight into that. The other reason, and this is possibly a little bit more contentious, so we point fingers at countries like the UK, which are busy meeting their responsibilities under 30 by 30 by closing their overseas territories to fishing. So places like St. Helena, Ascension, Pitcairn, Tristan da Cunha. And there's an aspect of ugly colonialism about that, largely in part because within the UK, for instance, they don't do a very good job of protecting the waters around the UK. So it is not fair to shift the burden for protection into these other places where there are fewer voters, let's just put it that way. But we also need to recognize that because remoteness is so important, particularly for pelagic species, as we're seeing in our results, that these countries are actually, these locations, territories, countries that are doing it well in remote places are actually providing an outsized service to the rest of us. And so there is actually a responsibility to think about how we support them in that activity because they are protecting what our data suggests are some of the most important places for pelagic species. And with that, I'll catch us up on some time and can take questions. Thank you. Oh, shoot. No, sorry. I got so busy about complaining about politics that I forgot my last slide. So this is basically what I just said. These remote big blue splotches on the map are incredibly important, and we need to keep our focus on ensuring that they get protected. Also interesting that Australia's only big, highly protected areas are also in our territories. So Cocos Keeling Islands and Christmas Island most recently. And then I just want to acknowledge this is a lot of data. We had a lot of great collaborations, a lot of great co-authors. I particularly want to give a shout out to the lead on our science paper, Tom Letessier, who was my postdoc. And while he was looking for a real job at a university, which he now has, and well-deserved, he just continued to work on this data. So I just also want, for the young people in here, want to remind you that discipline and perseverance is absolutely essential to being a scientist. And now I'll stop and take questions. Thank you.